Brilliant. Thank you, everyone. So it's a great pleasure to be here today talking about biomolecular simulations. So these are simulations basically of proteins, DNA, nucleic acids, sugars, these types of molecules. And I'm going to start with an invitation. So I am chair of this organization, CCP Biosim. And we do work actually very closely with CCP5 and many of the other CCPs. And what we do is we promote simulation to people who want to learn more about it, people who want to use it for their to complement their experiments. We run conferences, workshops. We have an industry series, which is pretty much focused on the pharmaceutical industry, but we're very, very open-minded to other contributions. And one area that might be of interest to this particular community is we do run every 2 years a multi-scale modelling workshop with CCP5, which is the CCP for materials. So maybe people might be interested in coming along to that. So that's on the 31st of March in 2025. So do sign up to our mailing list if you're interested in anything to do with biomolecular simulations. And if you're interested in other types of materials modelling and you don't know where to go, then between me and Chris, we'll know where to direct you because, so being chair of CCP Biosim, I have an overview of the sort of types of people who do what simulations in the UK, as does, of course, Chris. So I'll tell you a little bit about the sorts of things that I do. So I'm going to just play this movie. So this is a molecule that you can see wobbling around, and this is actually the mini replication transcription complex in the SARS-CoV-2 virus that caused so much trouble a few years ago. So this is the piece of molecular machinery responsible for making the RNA, making copies of the RNA from the virus. And of course, what you can see really is you can see this molecule moving around. You can see this, what you're looking at is the thermal fluctuations of the molecule. And our area is, I would argue, really quite mature. And that's because, as you will see shortly, it is used for drug development. So the conceit is, and it's not my conceit, I include the reference at the bottom, and you'll notice that my name isn't on there. So it's not my conceit, but the conceit of the field is that our methodologies, our technologies are so advanced, our force fields are so good. Obviously, they're not perfect, but they are pretty good. Our force fields are so good that it's as if we had a computational microscope that can enable us to look in atomistic detail at biomolecules and see the way they move. And obviously, it's very difficult to watch the motion of the thermal vibrations of a biomolecular system experimentally. So the way you can do that is through computer simulation. So how do these computer simulations work? Well, our simulations are completely classical in nature. So what we do is we label every of the atoms. So the atoms are treated as simple balls. The bonds between them are covalent springs. And we assign a force field. So we have bonds, angles, dihedrals. And then we sort of paint the atoms with some chemistry. So that's the sort of van der Waals interactions, electrostatic interactions. And all of these little parameters that you can see in this big equation down here, these are the force field parameters, which are determined empirically, or now what people are trying to do is derive them through machine learning, which of course turns out to be a lot more difficult than was anticipated. But it's going to work sometime. It definitely, it's one of those areas that shows a great amount of promise. So, of course, we don't just have our biological molecules. So you can see in the top right-hand corner, there's a little circular piece of DNA and it's surrounded by water molecules. So state-of-the-art simulations also surround their biological molecules with water and counterions. So one of the biggest application areas for biomolecular simulation is in the pharmaceutical industry, so for drug design. So again, what you can see, let me just get my little pointer. What you can see up here on the right-hand side, this green object, it's a protein from SARS-CoV-2. virus. It's again a molecular machine that helps in making new RNA for the virus to replicate. And you can see these little yellow things and these pink things and these purple things. So these are these sort of pockets in the protein of potentially where drugs might bind. And what we've done with our docking software is we've docked in potential pharmaceutical compounds into these sites. And then what we can do with our atomistic simulations is we can then do a sort of a more accurate calculation, if you like, to see if the molecules stick where the docking program put them. So we can try and use these sorts of calculations in small molecule design. We can, of course, also do this at the next length scale up. So if we want to look at how whole protein complexes hold together, or RNA complexes, or DNA nanotechnology, these are also calculations that are done in our field. So we're also very lucky in that, because we are to do with drug design, an awful lot of expertise, talent, knowledge, blood, sweat and tears has gone into the software that we use to run these simulations. So we have really well-established code and really well-established force fields that are developed across the international community that everyone uses. And there are, there's basically about 5 of them, and most of them run on GPUs. So they're really very sophisticated tools that we have available to us for these types of simulations. So again, so about the sorts of simulations that I like to run now. So I'm very interested in DNA, and have been throughout my career, because DNA is just such a beautiful molecule. So what I am trying to do now is switch my laser pointer off so that I can... okay, here we go. So what you're seeing at the top, you're seeing a simulation of a bit of DNA, again, changing shape due to thermal fluctuations. But you'll notice that this little bit of DNA is circular, whereas most of the time you think of DNA as being a linear sequence. So it turns out that your genome is not static. In fact, your genome continuously sheds bits of its DNA. And what bits of your DNA you shed are dependent on tissue, for example. And these extra chromosomal DNAs, they've been known for a long time, but they're getting a sort of bit of a resurgence in interest, basically because the technology that is available to detect them has massively improved. So you can see on the top right-hand side some, papers and journalistic statements about these extra chromosomal DNAs, these DNA circles. So these are really, really tiny ones. And the ones that I look at are very tiny, because my calculations are so computationally expensive that even with the world's biggest supercomputer, we're still really, really limited in the size and length of simulation that we can do, because we look at systems in so much detail. So I look at very small systems, and these things do exist. They are cut out of genomes, but some of them are very big. So some of them can megabases in size. This one's about a few hundred. And they can lead to cancer and all sorts of types of diseases. So the point is when we have supercoiled, when we have circular DNA that's not linear, it can actually be supercoiled. So it can be twisted or untwisted. So what I'm going to show you at the bottom here is a simulation of a piece of DNA that's been untwisted before we join the ends together. So here we go. So some of these circles that get cut out of your genome might be like this. So they might be supercoiled. And then the interesting thing about this one that I'm playing here, so this is a simulation of a negatively supercoiled DNA. And you're going to see, yep, there it goes. You can see that actually the DNA opens up and forms a hole in the duplex. Now, the reason that that's important is because the information, the genetic information within DNA is locked on the inside. So the DNA has to be opened by molecular machines in order for this information to be accessed. But if you've negatively supercoiled your DNA, you've pulled apart the van der Waals interactions, holding the bases together. And so you've weakened these base stacking interactions. And so your DNA can naturally sort of fall apart. So this means that the information, the genetic information contained inside, is easier to access if your DNA is circular and negatively supercoiled. And we think this might be important in determining what these ECC DNA circles might do, how they might behave biologically. So this is it. I wanted to show this work specifically because it's been done in collaboration with colleagues at the Royce. So I hope you can see at the bottom right-hand side here, these are the DNA stars. So there's me in the middle. So the real stars on the left-hand side and the right-hand side. So Alice Pine, who is an expert in atomic force microscopy, she's with the one with the hat on. And Agnes Noy, who was actually a postdoc at the time, she's got her own independent group now. She's the one on the right-hand side with the glasses, and she did these simulations. So Alice is so talented, and you can see one of her AFM pictures right in the center of this slide. So the stripiness, this is a DNA mini circle, pretty much identically to the one that you saw a simulation of. And the stripiness that you can see on this DNA mini circle is actually the helical repeat of the DNA, because Alice is so talented, she can actually see that with her AFM. And you might be able to see as well, labeled with a little red triangle, you might be able to see that there's a little defect, there's a little gap in the DNA. And that is exactly one of those defects that you saw forming the simulation in the previous slide. So what the DNA stars did in our calculation is Alice looked at the supercoiled DNA mini circles on a surface. And you can see the picture on the bottom left. And Agnes used molecular dynamic simulation, so that's the picture at the top with the little triangles on it, to model exactly the same simulations, exactly the same circles as Alice was imaging. And we were able to then use the atomistic models to interpret the imaging data. Because although Alice's imaging is better than anybody else's, and she gets better resolution than anyone else does on DNA for these circles, she still can't actually see where the atoms go. We can only do that with the simulation. So used synergistically, we can combine the simulations and the experiments and get more than the sum of the parts. And I noticed that very wisely, the Royce Institute has decided that imaging and characterization and simulations and modeling are the new theme areas. And of course, this work that we did together demonstrates that they go perfectly hand in hand. So now on to something a little bit different. So this is something that I've pioneered pretty much with other mathematical colleagues in Leeds. So I was in Leeds for many years. And what you can see here is two works of art. And these works of art show you an imagination of what the real cytoplasm inside a cell would look like. So the first thing you probably notice is that they're very pretty. The second thing that you notice is that they are very busy. And maybe the third thing you'll notice if you're an expert is that there are no atoms shown in these pictures. So these pictures will be far bigger than the biomolecules that you've seen. So one of the blobs in here will correspond to one of those proteins that I can model with my atomistic simulation. So you can quickly see that actually there's a need for something beyond. You know, if we want to simulate a whole cell, there are 10 to the 14 atoms in a eukaryotic cell. You know, we want to go from atoms and drug molecules all the way through to biomechanics, you know, what effect these molecules have on an organism. then we're going to have to do some sort of multi-scale modelling. And it so happens that at the sort of intermediate length scales, and what we call the mesoscale, so that's length scales of around 50 to 500 nanometers, there isn't really that many good modelling techniques for soft nanoscale objects. So that's what we've been trying to invent. And just to point out that, okay, so I've talked about atomistic modelling, and I'm talking now about mesoscale modelling, so the next scale up. But of course, if we don't have any chemistry, if we don't have any burning of ATP, of fuel, then we don't have any life, because you can't have any biology without fuel, without energy. So you know, actually we need the quantum chemistry as well. It's just I'm not talking about that today. Okay, so what we've done to try and plug this gap in our modelling expertise is we've cheated. So we've gone to the engineers and we've stolen finite element analysis. So finite element analysis takes a complicated 3D object and represents it using a lot of tetrahedron, and then solves Newton's equations of motion at various positions on the nodes of those tetrahedra. So we've stolen that, but of course what we want to do is we want to model proteins, and proteins are soft nanoscale objects. So the difference between the engineering applications and our nanoscale applications are thermal fluctuations. So I collaborate with a guy called Daniel Reed. He is a professor of noise. And so he took the conventional finite element analysis, and what he did is he added thermal fluctuations. So now our continuum equations of motion, so we've got, you know, acceleration here. So this equation basically says force equals mass times acceleration, and the force is on the right-hand side. So it's a divergence of an elastic term, so how squishy our object is, a viscous term, that's the fact that there's fluid, and this thermal noise. So what we end up with is a fluctuating jelly model of a protein that is in the continuum limit. So it's not a replacement for atomistic simulation. It looks a completely different length scale. So just to show you a little bit of the sort of workflow that you would do with fluctuating finite element analysis. So and I've got to undo my laser pointer again. You go to the protein database or an electron microscopy database, and you get your biomolecular structure from experiment. You then build a volumetric mesh of that structure. This is an absolute nightmare. So if there are meshers who want to talk to me about helping me build meshes, please get in touch, because it's a complete nightmare. Then, so this is the sort of texture of our matter. Actually, a lot of our simulations are overdamped. So this one does show inertia, which normally we don't have. But and then we end up, this is actually a simulation of the molecular motor dynein. And the molecular motor dynein is responsible for walking around your brain and carrying cargo. And these simulations were run in collaboration with cryo-EM microscopists at Leeds, who were able to see these things walking using microscopy. So again, this crossover between microscopy, which in our case is very often static pictures, and simulations where we should be able to add the, you know, sort of animate those otherwise static images. That's one of the aims of this project. So here's lots of acknowledgements for the DNA work. So we have a really wonderful community. If there are any early career researchers on the call, my biggest advice to you is pick a community that has nice people in it, because then you'll have a really fun career. And these are the wonderful people that have helped with the mesoscale modelling. And I'm really happy to answer questions, and I'd love to have new collaborations. So thanks very much for listening.